now that we go to the phenolic compounds in whole grain foods, and as you saw and know, whole grain foods are also the major source of dietary fiber in most diets. And this actually is very much about the associated compounds, which can be sometimes considered part of the fiber, or then they at least are in the same plant tissues. And let's see if this works. No, I have to change from. And if we think of the whole grain as it appears in the field, it has the cover of the grain, there's a germ which contains the starting part of the new plant, and then it has the endosperm, which is the starch and protein reservoir. And if we think that the dietary recommendations say fruit, vegetables and whole grains, the outer parts of the grains, which is the aleuron, is very similar to the let's say constituents which we might have in some fruit and vegetables, but the difference of course is that they don't have so much energy in them because they don't have starch. But anyway, that's the whole grain, and I will come to the thing why whole grain has been a very important thing, especially the word whole. It's because they started to learn some 15 years ago in epidemiological studies, like in the fiber story, that if you differentiate people who have been eating more of whole grain, actually in those epidemiological studies people were eating more bran or whole grain, you are protected. So you are protected for type 2 diabetes, and these are typically US cohorts, and people there eat very little whole grain altogether. So 2-day serving increase was about 20% reduction in disease risk for type 2 diabetes, and the similar lowering of risk in cardiovascular disease. There were many epidemiological studies, and these are now the meta-analyses which came after those, pulling together. And there are more studies coming all together showing that there's something in whole grain. There's also something in the cereal fiber. And that has led to recommendations and health claims especially in the US. So also the same dietary recommendations which I referred to in fiber, they say consume 3 or more whole grain servings, and at least half of your cereal servings should be whole grain. So then the question, what is in the whole grain? Again, when we start to process the grain, we should know what we should have from it, because nobody eats grains. They are quite hard and they are not almost edible as such. And there's a very good review, some 60-page review by Anthony Fardet in INRA, France, about different mechanisms and different compounds which are there besides dietary fiber. As we saw, the fiber, which is more the carbohydrate part, sure has some health outcome and is influential, but there is so much more. And I just recommend for you, this is one of the papers which you might be able to, if you are interested, to have a look. But I have some short descriptions here, and I'm going to concentrate on phenolics only. Phenolics is part of those compounds. But if we think that we have the energy compounds, and then we have the fiber in the grain, that we have known like 40 years. As I said, the fiber has been a topic. But now, in the last 10 or so years, we are all the time revealing new compounds which are there, which are not, if you quantify them, they are micrograms per gram, so not very big quantities. But the biological activity can be important, especially when we have many of them. Vitamins and minerals were known already earlier. Now we know of many others, and we know about their bioactivities. They are enzyme inhibitors, they are antioxidants. As we discussed, they can be phytoestrogens or they can be whatsoever. And now the search for these phenolic compounds and their effects is big. And this is the same study as for for berries and vegetables. There are big conferences on polyphenols and health. So whole grains are part of that story. And as I have to point out that refined grains are enriched with energy, and they are devoid of most of those compounds I mentioned here. And that's the majority of the cereal food in Europe we are eating is white. And just made a cartoon of that. If we think we have whole grain where we have energy and fiber and the phytochemicals, the phenolics, when we take the refined grain, we take the outer part of the grain away, give it to animal feed typically. We still have all the energy left. We have only little fiber and only little phytochemicals left. So that's what we are eating. And that's my big motivation, is to bring the fiber and phytochemicals back in the food and make it in such a form which you can eat it. And that's why I have coordinated also a big European program with 40 partners and 5 years on these aspects, where we learned a lot also about phytochemicals. And if we think what we have in the Anthony Fardet's 60-page review and what kind of compounds we have, this is what I already talked about, the fiber and the oligosaccharides. Then we have phytate, phytosterols, tocols, vitamin E, folate, the methyl donors, minerals, which all have some specific hypothesized activities, how they can contribute to health maintenance. But I now concentrate on these when I speak about whole grain polyphenols. It's alkylresorcinols, ferulic acid, and lignans. And here are some of the main activities. There are many activities, but the idea probably is that food is not a pill, and we cannot take ferulic acid from there and say, eat ferulic acid, that's good in the whole grain, or lignans. And there are all kinds of extracts being developed all the time, but the good thing about food is that we get all of it. So if we think of the phytochemicals, so the phenolic acids, they really are quite strong antioxidants. Ferulic acid is by far the most abundant, but also synapic acid is the next biggest. And as you will learn, the phenolic acids never are as they are always in the bound to the ferulic acid. Typically is part of arabinoxyl backbone, and they are bound to the matrix. So only a few % of the phenolic acids are as free in the matrix, which means that you need some esterases and enzyme activities to release ferulic acid so that it can be further metabolized in the body. Alkylresorcinols, they are phenolic lipids, and they have a fatty acid chain, and then they have the phenolic moiety. Depending on the fatty acid, they can be divided to according to fatty acid length. And as I will show, they have different bioactivities, but they also have been of specific interest because they are specific to whole grain, rye and wheat, and they could be used as biomarker of intake of these grains. And lignans are polyphenol metabolites, which are dimeric forms of cinnamic alcohols. And as I will show, they are metabolized by the intestinal bacteria in the same fermentation where we are carbohydrates and fiber are fermented to other types of lignans. And then they have been linked to different disease risks. This is research where I can tell you a story also, because I've been working with this a lot myself. Interesting science. I don't go to the others, I just say they are there. So remember, when we speak about whole grain phenolics, it's only part of the phytochemicals. And then I just say now I made a mistake, benzoxacinoids, it's so difficult word. There's at least N missing from here, benzoxacinoids. But anyway, I will also tell in my end of my talk that this is a very recently identified. Therefore, if you look at my slides, this was my old slides, 3 slides. Now I have the 4th slide. This shows how it's going, because this group was not known 5 years ago, or not realized. It was partly known, but now it's identified in wheat and rye grain. We have these. We also already now know they are absorbed and that they have some activities. So that's another group of compounds probably being studied a lot in the near future. All right, and then if we think of of the diet as more whole, the dietary phenolics, we now spoke about lignans and phenolic acids and acrylsanols, but then we also have flavonoids, and some flavonoids we can have in some grains also. Not in big quantities, but there are some. And then there are some other plant sources which are like berries or fruit which are really rich in some of the flavonoids. And of course we have phenolic acids in many more foods than in cereal grain. And then we have the complex polymers like L-gitannins and proanthocyanidins. So it's a big amount of chemistry in the foods, in the things which we can identify in foods. Okay, but when we go to people who have been eating these foods, and I have been in this research for, I don't know, 50 years. I remember very well when I think it was in 2000 in Salamanca in a congress of polyphenols and health. That was one of the first congresses where people started to, still then people were studying these compounds, and they were trying to do in vitro and other studies to show that what kind of bioactivities these compounds may have. And only in that congress people started to speak aloud, but why do we study these compounds? Because actually when we go to the circulating phenolics which we have in the body when we have eaten that food, they are not the same molecules anymore. If you think what are the metabolites of flavonoids, isoflavonoids, I will speak about the lignans, I will speak about phenolic acids. We rarely see the original compounds in the fluids, because they go, as I will show, they go to the transformations there, and then they go through the circulation. They are metabolized by the bacteria in the fermentation to other compounds, which then might circulate before they are secreted in the urine. And this is only, I would say, 10 plus years that this has been, let's say, very, very broadly realized. And I just want to say that my colleague Anna-Maria Aura at VTT has written one of the reviews available for about what happens in the gut microbial metabolism of the phenolic compounds. And we also wrote a review on rye phenolics, which are the same as wheat phenolics mainly. In Nutrition and Health we were concluding the literature. That was in 2009, and I must say that this is a very rapidly progressing area. So there's already quite a lot more research after that. Isabel Bondia originates from Spain and has been also working with Alfredo Martinez and is currently working at VTT in Finland, studying still partly these things. So again, going back to the grain and these compounds in grain, I just want to emphasize that they really are in the outer layers of the grain, which is called arleuron, and bran is a technical concept. So if you look at the antioxidativity, this is not the phenolic compound, this is the antioxidative activity measured from the flower. In the endosperm, in the inner part, there's quite small antioxidativity, whereas there's quite more in the outer parts. And in the same way, when we have looked at different milling streams of rye, these are the white flours. This is the whole meal flour. We have made it so that it's 100% O1. And then when you take the short, which is the intermediate fraction, and bran is the outermost fraction, you see that in the outermost fractions we have much more phenolic compounds than in the whole meal itself. And in the endosperm we hardly have. The phenolics are just in the outer parts of the kernel. The same is true for acrylsanols, lignans and phenolic acids. All of them are almost non-existing in the endosperm, but they are high in the bran. And this reference I quickly had there also, but as I said, there are groups, not only Soracalixte but others, who think that carrying of this type of compounds is a very important part of the grain fiber. And also in earlier studies, this was in 2009, they show that total phenolics and antioxidant potential, even though I must say, and I personally am not an antioxidant, a person favors studying antioxidant activity because it's so difficult and so broad concept. But anyway, that's what they saw, that when people were eating wheat bran, you could see that there is plasma uptake and urinary secretion of phenolics and increased antioxidant potential. But you can see that some of the materials in the food appear. And now if we think of the metabolism of these compounds, there are many sites, of course, in the body. There are some enzyme activities, and especially there's glucuronidase and sulfation in small intestine. Liver participates, of course, because these are partly xenobiot compounds, kind of foreign for the body, and that's the role of liver to work on these. But really the colon microbiota is doing all kinds of degradation. They're breaking down the phenolic rings and substituting and so on. And then they, from the large intestinal fermentation, they go to the liver and in the enterohepatic circulation, and they actually, there's a continuous change actually after, if we take in a lunch something very polyphenol rich, it can also be wine, of course, and then so it starts to be modified and circulated, and then the clearance is then via urine, and depends very much on the quality of the type of the metabolites, how long they would stay in the circulation. But not, this is a very long time. Some compounds already are quite quickly secreted and disappear from the circulation. And if we think that, another picture of the same, that we have the polyphenol, typically they are as glycosides. They quite seldom are as agglutins, so only polyphenol. It's typically a polyphenol, and then there's a sugar moiety or some carbohydrate attached to that. So they first need to be cleaved off, those sugar moieties. Then we get the polyphenol agglutins, the pure polyphenol part, and then we have the colonic bacteria working with the serine fission or other modifications, and then we get metabolites which are absorbed. Some of the compounds can be absorbed as such first. So some compounds appear in the circulation, but most, as I saw, go via their metabolites. And it gives really big complexity to this because we have many phenolic constituents, and then we have many metabolites, so we have an array of compounds. But in the same way as I showed, and as I said, I'm interested in the diabetes risk and in the insulin sensitivity, inflammation, and that's why I'm interested both in fiber and the polyphenols, because if we think that people are eating this food which always contains both fiber and polyphenols, and as I showed, this is what we can have as from the fiber. Fiber can change the microbiota, but also polyphenols can selectively change microbiota, because some polyphenols are really strong antimicrobial compounds. And actually that has been more studied in berries, but it has been shown that some berry polyphenols, they suppress the growth of like pathogenic bacteria, but they are not suppressing the growth of lactobacilli and some, let's say, more favorable bacteria. But the most important thing is that there is the release of phytochemicals, which then go in circulating phytochemical metabolites. And then if we are studying adipose tissue function, for example, they have an influence. I will show to you. Alkyl resorptionals have been shown to have an influence on the lipolysis and lipolysis in the formation of free fatty acids in the adipose tissue. So then they have in the target tissues, they interfere with the reactions. And by that way they can then influence the inflammatory status or insulin sensitivity, and in the long run contribute to the risk of diabetes, for example. I have a student, Jenny Lappi, who just wrote a review on the role, let's say, on the role of fiber and the phytochemicals, on the mechanisms. All right. Now we then go to the distinct groups of compounds. The most abundant is the phenolic acids quantitative wise. And if you look at the, I always have my examples of rye because our national whole grain cereal bread in Finland, but they look the same if you take wheat, for example, which is the national whole grain food in most other countries. And this is the outer part of the grain. Here we would have the endosperm, here we have the aleurone layer, and here we have the pericarp. This is mostly lignin and cellulose. And here we then have all the phenolics are really, as I showed to you, this is the layer where they are. And if you look at the phenolic acids, the majority is ferulic acid, and the majority of them is matrix bound. So they are part of the cell wall, which I showed to you, this insoluble complex mixture. Then another part is conjugated, soluble there in the grain, but they are conjugated to carbohydrates and other compounds, and only a small majority is free, and very much of the synapic acid actually is then free. So that is what we have there. And then when we eat this type of food, first the ferulic acid must be released from the matrix, and that is known to happen both in the small and large intestine by the esterases which are present there. And when we have the free ferulic acid, then that will be then converted first to hydroxyphenylpropionic acid and then phenylpropionic acid. And then they are glucuronidated and sulfated because they otherwise will not be able to so well get along in the mixture which blood is, and then they finally are excreted in urine. So that is typically what happens to ferulic acid. The interesting thing is, and there is one PhD work just going on in Montpellier in collaboration with us, where they prepare different types of wheat brands where the ferulic acid was more and more, let's say, liberated from the cell walls. In extreme case was pure ferulic acid, but that's another story. But then they had solubilized polymers where ferulic acid is there, or then they have hydrolyzed polymers with ferulic acid attached, and then they had released ferulic acid. And now we have done a series of experiments within in vitro fermentation, also animal experiments, to see how this degree of ferulic acid attachment to the food matrix influences its bioavailability and the metabolism in mice and in vitro. So it's very much about the food matrix effects, partly in the first part of the absorption, before we can come to this type of metabolism. And what we showed also in two papers, this is part of the health grain project and a collaboration. It was actually also a Spanish girl, Nuria Mateo Anson, who was working in Maastricht and in TNO, and worked with bran. We did some bioprocessing of the bran at VTT, did some in vitro work in this case in the Netherlands, and looked at the fate of the phenolic compounds. And we see that we wanted to see if we can improve the availability of the phenolic acid by bioprocessing. That means we were fermenting and using enzymes to pre-treat the bran. And the idea is that when we have the complex cell wall matrix, if we partly degrade it before we have it eaten, it would facilitate the metabolism of the phenolic compounds. And in TNO there are these TIM models, TIM 1 and TIM 2. This is the small intestinal model. And you can see that the bioaccessibility of ferulic acid was increased. This is always this is the bar where we had the bread with bran which was fermented and enzyme treated. This was only fermented. Here is just whole wheat bread, and this is white bread, and this is whole wheat bread. So you see that actually not very much was liberated from even though there was quite a lot, it wasn't liberated. But when you treat it with enzymes prior to eating, you liberate much more. And then what we saw, if we put it in the colonic model, in the so-called TIM2 model, again the same bar, so that the last bar here is the one with the enzyme-treated bran, and here is the white, and this is the whole wheat bread. You see that when you look at the bars, that the phenolic acid is released, and because it already was released, there is less release actually here already in the first phases, because partly it was released. And then when we look at the formation of the metabolite, we see that much more metabolite was formed from the two bioprocessed brands than from the others. And actually we also showed that the metabolite, let's say the dynamics was different. So you not only have different amounts, but you have different ratios of those metabolites, which I showed for phenolic acid in the previous picture. So by just having the same food, composition is completely the same. But because we pre-treated the matrix before in the food processing, we increased the release of the phenolics. And later we showed that when we made a human study then with this type of bread, we again saw that the phenolic acids were more bioavailable, but also when we then did ex vivo studies with some anti-inflammatory factors from the blood, we saw that they were also increased. So this shows that food structure engineering or food processing is important, as it is important for the bioavailability of the nutrients. We know very well that we can create resistant starch, and we can retard the glucose absorption, and we speak about rapid and slow proteins in terms of digestion. We can also modulate the metabolism and uptake of phenolic compounds in the food. And of course, some of them go through us. We can recover some of them in feces. And some hypotheses are that the local effects in the gut are very important as well. So even though they would go through us, they would already be important for the colonic health, for example. But of course, for systemic health outcome, the ones which are absorbed are important. Then we go to the phenolic lipids, alkyl resorcinols, and there is a lot of research about their use as biomarkers of intake. And of course, biomarkers of intakes would be really useful if we are looking at the compliance of intervention studies, but especially because in epidemiology and in population level and follow-up studies, we only have to rely on questionnaires about what people have been eating. And if we could have a specific biomarker of intake, it would bring us to another level of sensitivity in linking intake on some food and health outcomes. And especially in Sweden, in Uppsala, there have been a lot of this research, and Alasdair Ross, who has now long been working in Nestle in Switzerland, but he just recently, and he did his PhD in Sweden, he just this year wrote a review article about the status and perspective of the use of alkyl resorcinols as biomarkers. And this is from his review. He has there from different papers collected the relationship between the alkyl resorcinol intake as part of the intake of whole grains and the plasma total alkyl resorcinol content, and you can see that it's rather linear relationship between those two. And they are well absorbed, but then he's saying that if there are very big quantities eaten, then it might be that not all of it is absorbed. And also there is work about their role in the lipoproteins, like HDL. There is one PhD done in Finland on that. Another one will be defended in November about alkyl resorcinols and their role in lipid metabolism. And also that, I will come back to this adipose tissue. And then they are metabolized like most others, to two different metabolites which also can be identified and can be summed up if we discuss the biomarker question. And this is the paper from University of Lund, also in Sweden, and you see that we both in Finland and in Sweden, we really study a lot the compounds in rye, because we eat so much rye bread, but also because rye is very abundant in these compounds. And they are interested in diabetes, and they are saying that reduced lipolysis and lower levels of circulating free fatty acids, that can be one of the reasons behind the lower levels. And that is what we have seen quite often in postprandial studies, in the late postprandial phase, less free fatty acids being formed. So alkyl resorcinols are interesting from many points of view. And then I come to lignans, and as I said, this is where I feel I'm part of the history of the lignan research, partly because, or not partly, completely because I was working with Professor Herman Adlercreutz, who is now just had his 80-year anniversary seminar, but he's really the Mr. Phytoestrogen. He's been working with phytoestrogens all his life. I don't know, has some 600 papers probably on them. And as he's working in the same city and we were working in the same Nordic project, we started to work with the lignans in rye. And he's the person who about 25 years ago already had the hypothesis that there is not only fiber, there must be something else and that it's the joint action of this type of compounds and fiber that the health outcome is reached. And at that time, the interesting story which I can tell you is that at that time we only knew that these are the two lignans which are present in cereals. So mataresinol and secula-isoresinol. The most abundant source of lignans which we know in the diet is flaxseed. Flaxseed contains something like, I don't know, 1,000 times more secula-isoresinol than the whole grain cereals. So if you want to have a really big portion of lignans and secula-isoresinol, please eat flaxseed. Okay, but we knew these two, and we knew that they are forming enterolactone, first enterodiol, which is then converted to enterolactone, and that is the mammalian lignan, and that has got phytoestrogen activity, and this is the compound which is of interest in terms of breast and prostate cancers. And there's lots of research in both in Canada and for breast cancer, and in the Nordic countries for breast and prostate cancer with respect to mechanisms of these compounds. But the interesting thing in science was that when we were measuring enterolactone levels in human beings who had been eating cereals, and we were also doing these in vitro studies, we were fermenting grains with measured amounts of mata and secula-isoresinol with gut bacteria, and we're measuring enterolactone. We always had almost 5 to 10 times more enterolactone than we could analyze these compounds. And we were wondering, what is the problem here? Can't we quantify how much we have these, or is there something else? And then, I don't know if I have it here, yeah, perhaps I don't have the reference, but about 10 years back, then we eventually understood that there are many more lignans in the grains than we thought, not only these two, but we also then realized others, and we got standards from Japan for each of these compounds, and we incubated them with this in this simulated gut fermentation. And then we realized that many of them produce enterolactone. They metabolize to each other and then further to enterolactone. Not all of them. There are some others which are not metabolized to enterolactone, but some others are. And this is the same picture which I showed to you, but now, so in the gut fermentation by the bacteria, there is this deglycosylation, there is this transformation and conjugation, and we get enterolactone. And what we then, this is the reference to the paper which has been really highly cited, because then we showed that to which extent each of these lignans are converted to enterolactone in vitro fermentation. So now we know still more about lignans already. We also know that that there are even more lignans, and that there are, I will show you in my last slides. We just realized there are polymeric lignans also in rye, and we don't know if some of these are circulating in the body as such, but mostly they are circulating as enterolactone. And this is the last part of the talk, and then I go to this current research we have, which is very much based on the non-targeted metabolic profiling. And these are, it's my colleague Kati Hanheva and Isabel Bondiabons, VTT, and she's at University of Eastern Finland, who are working with this. So we are studying both the breads and grains, and then the human fluids. We are analyzing them in the metabolomic HPLC-MS system. Then we do the data analysis. We identify metabolites when possible. As I will show to you, even though we don't always we can't identify all of them, we can see that how big changes there are in the metabolite pool, and then we try to interpret what's going on in the physiology. The first thing was that when we took the major grains grown in Finland and did just from the extract this type of phenolic fingerprint analysis, we could see that they are really different from each other. This is one of the clear things that, like I showed, and we know that dietary fibers are very different. Even though all whole grains are probably better to eat whole grain than refined grain, the grains are different in the fiber content. They are also different in their phenolic acid profiles. Like, for example, you were asking about oat. Oat is very rich in aventantramides, for example, and that's another group of which is specific for those. And these two contain at least alkyl resolutions and benzoxacinoids, which are specific for that. Barley has not so much been studied so far in this way. And we have, as I said, we have identified these benzoxacinoids. I had completely misspelled, as you see. So I can already say very well benzoxacinoid, but it's really bad word. I wrote it in hurry wrong. But anyway, we found them, and now there are two papers already, one from Denmark and one from somewhere else, where they have shown that this can be found also in biological fluids. So this is a compound where we will learn more. And we found this using this technique, so we've just screening for big metabolites in the rye extract, which were so far unidentified. The other thing which we find was these new lignans like butanol and so on, which are polymeric lignans. So they are much larger molecules than the lignans with just two phenolic rings, which we knew before. So this is showing that 10 years back we thought it's 2, then 10 years ago we learned it's 6, and now we know there's also polymeric ones. And after 10 years probably we know again something more when we are able to detect things. And I already been pointed out, but what we really are doing is that we have, like in the Holland they have these team models. We have at VTT our in vitro models. We have the enzymatic digestion model, which is simulating the upper GI tract, and then we have the human fecal inoculum, anaerobic conditions. We are fermenting, and then we study the metabolite pool, and then we try to link the changes we have there to health outcome and other things. And we just have now studied, and which is now in press in PLOS, the in vitro fermentation products of 2 samples from rye bran. We had both the soluble extract and then the very insoluble residue. Both contain phenolics. We had both go through this in vitro fermentation, and then we we analyzed the metabolite pool. We did not yet identify many of the compounds, but we could show that almost everything was changed. So that after the simulated colonic fermentation, you have clear clusters of compounds, and you could also in a PCA plot you could very easily get cluster. You could see that the compounds were typical of, for example, you could differentiate between the different substrates, where they were either more bound or less bound, and then you could with the different time points. And now we would like to go further to identify what all new metabolites. They are not all phenolic metabolites, because in rye-bran phytochemical pool, as I showed you, there are many other things than phenolics. But what are the compounds which are formed, and what is their relevance to the health outcomes? So this is the big interest, is that it's not only the food, it's the fermentation pattern. Then when we can have potential markers for bioavailability from the intervention studies, then we can go and we can get some ideas of what type of compounds or what type of mixtures are circulating. We can go to mechanistic studies in animal models, for example, where we can then sample well all the organs. And actually that is what we are also now currently working on. We are, after feeding trials with mouse, for example, with differently processed rye-bran fractions, we can collect all the tissues, and we can study also the tissue levels of these compounds. And that's the critical thing, because in blood we have the circulation, and it's kind of like, if you think visually, I think that if these are different cities or places, and then we have the blood which is circulating there, some compounds might be more at higher levels here and others here, and the site of action can be here or here. And now that we can, through metabolomics, start to detect the compounds in those organs, then we can really start to go for mechanisms, how the phytochemicals will influence the metabolism. And then, of course, we can go to cell cultures, which is a simplified system for some compounds, and try to see some of the mechanisms. So this is a holistic approach of how to try to understand the synergistic role of the many phenolic and other compounds which we have there. Because the difficulty in all nutrition research, as well as here, is that it's not only one compound, it's not only resveratrol, and it's much more. And well, I don't go, you can't see anything anywhere, but here is here we have just identified, and there are papers coming out identifying many of the metabolites. Some of them are direct phenolic metabolites, and others are already next stage metabolites, which are part of the pathways which then are linked to different disease outcomes. So this type of research is the one which is going on not only in our labs but in many others, and there are quite much data and also new reviews coming out a lot, as with fiber. All right.